Hello class, today I am presenting you the autonomous nervous system and special senses. So where is the place of the autonomous nervous system in the classification of parts of the nervous system? This is the classification of the nervous system we saw in the previous module. Look, the nervous system, if you put nervous system here, would be divided into central nervous system and peripheral nervous system. As we saw before, central nervous system consists of brain and spinal cord. Peripheral nervous system consists of all nerves which take impulse away from the brain and spinal cord, or which bring impulse to the brain and spinal cord. Those which bring impulse to the brain and spinal cord are known as sensory or afferent, whereas those which take command or order away from the central nervous system are known as motor. These motor ones are the ones which are divided into somatic and autonomic nervous system. Here I want to tell you what is somatic nervous system about and what is autonomic nervous system about. Somatic nervous system practically is controlling the skeletal muscles. Skeletal muscles are the organs which we have willful control over. So the somatic nervous system consists of controls the organs over which we have willful control. We can contract our muscles when we want to, and we can relax them when we want to. So that's what is done by the somatic nervous system. Whereas the autonomic nervous system controls organs over which we do not have willful control. Example, digestive system. The food we ate an hour or two ago is digested despite our will. We don't have willful control over when it is digested, how it is digested. It is controlled by the autonomic nervous system despite our will. Glands, for example, which produce hormone, which are in our body, they do produce the hormone despite our will. We don't apply effort for the food we ate to be digested and for the glands to produce hormone. These are just examples. We can come up with multitude of examples. The autonomic nervous system, which is our topic today, is divided into sympathetic and parasympathetic division. So the next question is, since the autonomic nervous system controls organs which function despite our will, how do these two branches or divisions of the autonomic nervous system differ from each other? Here is how they differ. First of all, most of our organs in our body, internal organs, have sympathetic and parasympathetic nerve attached to them, meaning we describe this as they have sympathetic and parasympathetic nerve supply. That means they are controlled by both. The effect of sympathetic division and parasympathetic division on individual organs is opposite. They act opposite. Let me show that on example. For example, sympathetic division, when sympathetic division is active, or when sympathetic nerves stimulate our heart starts to beat faster and stronger, starts to contract stronger and to beat faster, more times per minute. Whereas when the parasympathetic division is active, or when the parasympathetic division stimulates our heart starts to beat slower and weaker. This is the opposite happening on the same organ when these two branches of the autonomic nervous system stimulate. Let's take another organ, for example, our airways. Our airways are, for example, the bronchi, which we didn't learn yet. The airways which lead to our lungs are tubes through which air flows when we breathe in and out. So when sympathetic division of the autonomous nervous system stimulates our airways, the airways widen, broaden, and they allow more air to go in through them. Whereas when the parasympathetic division stimulates the airways narrow down. This is how opposite they are. And I want to tell you, most of our organs, majority of our organs, are supplied by both, and these two have opposite effect on those organs. This is not the only difference they have. As we go through this lecture, you will see more difference between the sympathetic and parasympathetic division of the autonomous nervous system. You will see more differences, and let's go for that. The other difference is they have difference in the neurotransmitter they use. They have difference in the structure of their and the route their nerves take. So those are the differences. I was telling you that most organs are supplied by both. That means if I say most, there are exceptions. So I would like to mention the exceptions. The exceptions are sweat glands. If you remember from our skin topic, integumentary system, the arrector pili muscles, and our kidney. So if we say they don't have both, so which one do they have? These three organs I mentioned, which have only one supply, they have only sympathetic. They don't have parasympathetic. Which organs? Our sweat glands, arrector pili muscles, and the kidneys have only sympathetic. Otherwise, most organs, except these three, have dual innervation, meaning double innervation, sympathetic and parasympathetic, being influenced in opposite way. So we will see more differences. I mentioned already the structure of the route of their nerves, the location of their ganglia, the neurotransmitters they use. Those are different. We will see as we proceed. Let's move on. Here is a picture which will show us the structural difference between the somatic and the autonomic nervous system. Let's see that also before we immerse into the autonomic nervous system. Let's see some of the differences between the between the autonomic nervous system and somatic nervous system. As I said, somatic nervous system controls our skeletal muscles. That's why you see this here. Somatic nervous system has neurons which extend from the central nervous system all the way to the muscles. I hope you remember the neuromuscular junctions is a connection between the tip of a neuron, the axon terminal of a neuron, and the skeletal muscle. And the neurotransmitter used at those junctions is acetylcholine. I want you to notice that. At all neuromuscular junctions, the neurotransmitter used is acetylcholine. By the way, this is abbreviation for the neurotransmitter acetylcholine. I want you to know that neurotransmitters are very important. As you have seen, neurotransmitters stimulate receptors on the postganglionic neurons. The receptors on postganglionic neurons are not the only ones which are stimulated by the neurotransmitters. Receptors on the skeletal muscles are also stimulated by neurotransmitter acetylcholine. So wherever neurotransmitter is released, there is a receptor for that neurotransmitter, and that neurotransmitter stimulates the receptor on that organ and brings about change in the function of the organ. So get that. Neurotransmitters are multitude. We are going to learn some of them here, and I want you to know neurotransmitter is very important. To show you, just to show you the importance of neurotransmitters, deficiency of neurotransmitter can cause serious nerve disease. Excess neurotransmitter can also cause serious nerve disease. So that's how important neurotransmitters are. Just to give you an example, have you heard about the disease schizophrenia? Schizophrenia is caused by excess neurotransmitter dopamine. Have you heard about Parkinson's disease? Parkinson's disease is caused by deficiency of the same neurotransmitter dopamine. This is how important they are. Excess is abnormal, deficient is abnormal, it has to be the right amount. So this is just to show you how important neurotransmitters are. Otherwise, you will not be tested on schizophrenia or Parkinson's disease. So that was about somatic nervous system. Here we have the autonomic nervous system, sympathetic, parasympathetic. What do we see about sympathetic division here? First of all, I want you to notice this ganglion. Ganglion singular, ganglia plural. Ganglion, as we defined it before, is a collection of cell body of neurons or soma of neurons. There where there is soma or cell body, expect neuronal synapse, right? That's what you see here. Here is axon terminal, here is cell body, there is synapse, and in the synapse neurotransmitter being released, right? So ganglia are part of the autonomic nervous system. There are ganglia which we are going to learn. There are several ganglia which belong to the autonomic nervous system. So ganglia are part of them. So neurons before the ganglia are known as preganglionic neurons, and neurons after the ganglia or whose cell body is in the ganglion are known as ganglionic neurons. It is the ganglionic neurons which extend all the way to the individual organ. What you see here is organ of digestive system, organs of the digestive system, which go all the way to the organ and have synapse there. The synapse here is no more neuronal, it is neuro-organ synapse. Like the neuromuscular junction, there will be axon terminal organ junction, where also are receptors, where also neurotransmitters are released. So within the autonomic nervous system, expect ganglion and expect preganglionic neurons, the cell body of which is located in the central nervous system. Do you see? Central nervous system? The cell body of this preganglionic neuron is located in the central nervous system, meaning in the brain or in the spinal cord. But the cell body of the second neuron, which we are going to call as ganglionic neuron from now on, is located in the ganglia of the autonomic nervous system. We will name the ganglia. We'll give name to the ganglia. This is showing you a general schematic anatomy of the autonomous nervous system compared to the somatic, and some comparison between the sympathetic and parasympathetic divisions of the autonomous nervous system. So the ganglionic neuron extends all the way to the organ and has synapse. What are we shown here? By the way, this is part of the sympathetic division, right? What are we shown here? What are we shown here? This triangular organ is the adrenal gland. This organ, which has a shape of a triangle, is adrenal gland. Adrenal gland is a gland located above our kidneys, one on each side, right and left adrenal glands. And this gland has a layer, innermost layer, which is known as medulla, adrenal medulla. The adrenal medulla plays important role within the sympathetic division of the autonomous nervous system. So the innermost layer of the adrenal gland, which is known as medulla, plays role in the sympathetic division. So we can consider it part of the sympathetic division of the autonomous nervous system. Its role is secreting epinephrine and norepinephrine. The adrenal gland secretes neurotransmitters or hormones, you can call them any way you want, which are released into the bloodstream. That's why you see a blood vessel here. This adrenal medulla, which is in the center of this triangular organ or gland, release their product, which is epinephrine or norepinephrine, into the bloodstream. And that, carried by blood to all parts of our body, they cause generalized change, which we are going to discuss in a minute. So I want you to notice that the preganglionic neuron has synapse with the adrenal medulla, and the adrenal medulla is stimulated by the preganglionic neuron. Okay, that was about the sympathetic division. So this triangular organ, which I named as adrenal gland, has a central part which is medulla, and that medulla of the adrenal gland is part of the sympathetic division. Let's move on to the parasympathetic division. Parasympathetic division also has preganglionic neuron, whose cell body is located in the central nervous system. It extends all the way to a ganglion, where it has synapse with ganglionic neuron. The ganglionic neuron, like that of the sympathetic division, extends all the way to the individual organ in our body and makes synapse. So I want you to notice some common things. Did you notice that the neurotransmitter used at the neuromuscular junction is acetylcholine? Likewise, I want you to notice that the neurotransmitter used at the ganglia, both in sympathetic and parasympathetic division, is acetylcholine. This makes it easy for you to remember, right? Neurotransmitter used at neuromuscular junction, at ganglia of the sympathetic division of the autonomous nervous system, at the adrenal medulla, and at the ganglion or ganglia of the parasympathetic division is acetylcholine. But when we come to the ganglionic neuron organ synapse, then the sympathetic division uses norepinephrine, whereas parasympathetic uses acetylcholine. I want you to notice that also. I also want you to notice the location of the cell body of the preganglionic neurons and the location of the cell body of the ganglionic neurons. Okay. And organs controlled by the autonomic nervous system. By the way, the effect of these neurotransmitters on individual organs could be stimulatory or inhibitory, excitatory or inhibitory. By now you know what is meant by stimulatory or inhibitory, because we saw this in the previous module we had. Let's move on to this one. What do we have here? What we have here is the anatomy of the parasympathetic division and sympathetic division compared side by side. They are compared side by side. Let's start with the parasympathetic division. I want you to notice that nerves which belong to the parasympathetic division are either cranial nerves, meaning some of those 12 cranial nerves which we studied, and some of the cranial nerves which we studied, and some sacral nerves. To name them, cranial nerve 3, oculomotor nerve, cranial nerve 7, facial nerve, cranial nerve 9, which is glossopharyngeal nerve, and cranial nerve 10, which is vagus nerve, are parts of the parasympathetic division, and sacral nerves S2, S3, and S4 are parts of the parasympathetic division. Because of this, they call the parasympathetic division craniosacral control. Obvious why they call it, because the cranial and sacral nerves are parts of the parasympathetic division. That's why they call it so. By the way, this part here represents ganglion. I want you to notice that. This part represents ganglion. I'm going to show you another picture which will make it clear that this is ganglion, but for now I want you. Do you notice that all organs on this side innervated by the parasympathetic division are the same as all organs innervated by the sympathetic division? This reflects the idea of dual innervation, double innervation, meaning all of these organs listed here have sympathetic and parasympathetic supply, as you can see on this picture, right? So I told you these are the ganglia of the parasympathetic division, which are really close to the organ. Whereas look at the ganglia is plural for ganglion. The ganglia of the sympathetic division are located close to the vertebral column, close to the spinal cord. So you can say this is preganglionic neuron and this is ganglionic neuron. Preganglionic neuron means a neuron before the ganglion, which is this. And this one is ganglionic neuron because it's after the ganglion and its cell body is in the ganglion. So do you notice that the postganglionic neuron is shorter than the preganglionic neuron? You need to notice that. Whereas here, look, this is the preganglionic neuron and these are the postganglionic neurons. Do you notice that the postganglionic neurons of the sympathetic division are longer, whereas the preganglionic neuron is shorter? That is because the ganglia of the sympathetic division are located close to the central nervous system, close to the spinal cord. These are the ganglia. They call this chain of ganglia as sympathetic trunk. They call it sympathetic chain or sympathetic trunk. It is a chain of multitude of ganglia located next to the vertebral column. And these are the postganglionic neuron. There are additional ganglia. These ones are known as paravertebral ganglia because they are located side by the side of the vertebral column. These ones are known as prevertebral ganglion because they are located in front of the vertebral column. There are additional ganglia here. There where you see such a picture, that's ganglia. There where you have neuronal junction, there is cell body, a bunch of cell body, then it's ganglion, right? So there are two groups of ganglion in the sympathetic division. Paravertebral ganglia, para means besides, ganglia located besides the vertebral column, and ganglia located in front of the vertebral column. These ones, don't disregard the location of this here. These ones are located in front of the vertebral column, so they call them prevertebral. Pre means in front of. Prevertebral ganglia here, paravertebral ganglia here. The other name for paravertebral ganglia is sympathetic trunk. These ones also have names, and I will tell you when we get there. These ones also have names. And I want you to notice that nerves which belong to the sympathetic division arise from the thoracic and lumbar region. These are spinal nerves which arise from the thoracic and lumbar region. Because of that, they call the sympathetic division thoracolumbar control, because it consists of nerves which arise from the thoracic division of the spinal cord and the lumbar division of the spinal cord. So this is a good comparison in terms of anatomy between the sympathetic and parasympathetic division. Let's have a look at this one. This is to show you the parasympathetic division separately. As I told you, do you see cranial nerve 3, 7, 9, and 10s, and then sacral nerve second through the fourth are parts of the parasympathetic division. By the way, these yellow colored parts are ganglia. I want to tell you right here and right now that the ganglia of the parasympathetic division are located close to the organ or sometimes inside the organ. Inside the organ. Do you notice that the ganglia in the liver is inside the liver? So that's why the preganglionic neurons of the parasympathetic division are longer, whereas the postganglionic or ganglionic neurons are shorter. So we have already said the neurotransmitters used at this ganglia, at the neuronal junction or neuronal synapse, is acetylcholine, and also the neurotransmitter used at the neuroorgan synapse is also acetylcholine. So the neurotransmitter used within the parasympathetic division is acetylcholine. So throughout, that might make it easier for you to remember. So the information we get from this picture is that nerves of the parasympathetic division are cranial nerves listed here, sacral nerves listed here, and the preganglionic neurons are long. They go all the way to the organ itself or to a ganglion located next to the organ. That's why preganglionic neurons are long, ganglionic neurons are short. By the way, this picture is to show you the location of the paravertebral ganglia or sympathetic chain. Do you see this ganglia? Two of them here, and the prevertebral. So this is spinal cord, which is inside the vertebral column, right? Isn't this in front of the vertebra or the spinal cord? Isn't this besides? So this is part of the paravertebral ganglion chain or sympathetic chain, and this is part of the prevertebral. They also call it collateral ganglia, one of the collateral ganglia, which are located in front of. What did we say ganglion is? Ganglion is a bunch or collection of cell bodies where there is synapse, right? That's the purpose of this picture, to show you the relative location of the paravertebral ganglia or sympathetic chain. This is a chain which goes really through the whole length of the vertebral column on both sides. This is shown just on one side. There is on this side also. So this is here, and these ones are in front of the vertebral column. What we see here is spinal cord, but you know that the spinal cord is located inside the vertebral canal. So you would expect the vertebra around here, right? So prevertebral or collateral, paravertebral or sympathetic chain ganglia. Here is the sympathetic division anatomy. As I said, this is a sympathetic chain ganglia. These are the paravertebral or collateral ganglia. The position, this doesn't reflect reality. It's just to show you how they are structured. The real position, the relative position of this ganglia and this ganglia is well shown on this picture which I just showed you. So this is the real position, but this one is just to give you idea, because the ganglia are located close to the vertebral column. The preganglionic neurons are short, postganglionic neurons are long, and all organs which are innervated by parasympathetic division of the autonomous nervous system are also innervated, are also supplied by the sympathetic division. And I want you to notice that the nerves which are parts of the sympathetic division arise from spinal nerves in the thoracic and lumbar division. Do you see those which belong to the parasympathetic division are not here? So do you notice that sympathetic and parasympathetic division have separate routes. They don't share spinal nerves, right? I want you to notice that also. So this is sympathetic chain, a chain of ganglia, or paravertebral ganglia. Is prevertebral ganglia or collateral ganglia. So in this ganglia, at the neuronal synapse, the neurotransmitter used is acetylcholine. But at these neuroorganic synapses, the neurotransmitter mostly used is norepinephrine. Mostly used is norepinephrine. This is where the difference between sympathetic and parasympathetic division comes, because in parasympathetic division we said the only hormone, the only neurotransmitter used is acetylcholine, but here acetylcholine at the ganglia, norepinephrine at the ganglionic organ junction or synapse. Okay. Okay, using this picture, I want to talk about physiological differences, physiological details of the autonomic nervous system. And I want to show that on an example. For example, when we are really excited, frightened, or in the fight or flight state, when we are in danger, where you have to fight back or fly away, escape a danger, right? When that happens, it is the sympathetic division of the autonomous nervous system, which is managing, which is in control, which is activated during fight or flight state, when we are really frightened, when we are really excited or in danger. So how does the fight or flight state go? So we said the adrenal medulla is part of the sympathetic division, right? When the sympathetic division is activated, the adrenal medulla will be stimulated. So when it is stimulated, it releases norepinephrine into the bloodstream. Norepinephrine and epinephrine. Epinephrine, by the way, is another name for adrenal hormone, the adrenal hormone or adrenal neurotransmitter. For adrenaline, epinephrine is another word for adrenaline. Adrenaline is a hormone also. It can also be used as a neurotransmitter. So this adrenaline, which is released into the bloodstream, is distributed to almost all organs. What does it do? It mobilizes all organs which have role in fighting an enemy danger or in flying away. For example, when adrenaline is released and at the same time neurons of the sympathetic division fire, for example, our heart starts to beat fast and to pump stronger. That will help us to run away or to fight. When our heart beats faster and pumps more blood, then our muscles will get oxygen and nutrients like glucose, which they will use to work, right? To really contract and fight or fly away. So, and also the adrenaline mobilizes our metabolism. For example, fat which was in storage will be released to be used for the production of ATP. So energy supply will increase, right? Any organ which would help will be activated. Any organ which would help in fighting an enemy or in flying away, escaping from an enemy, will be activated. What else? Muscles will be activated, right? That's a separate thing, because muscles will be provided with sufficient oxygen, sufficient glucose, so they may function. For example, our eyes, our pupil will be widened so we see well. Our brain will become vigilant to really think of alternatives, how to fight or how to fly away, right? So all organs which have role in fighting back or escaping will be activated. But those organs which do not have role, like the digestive system, doesn't have, will be suppressed, will be inhibited. Digestion stops. Production of urine is not important by then. Kidney can be inhibited. So energy will be mobilized to those organs which have role in fighting an enemy and escaping from an enemy. So this way, the sympathetic division of the autonomous nervous system, which is activated when we are frightened or in danger, will help us fight back or escape a danger. So fight or flight is a task accomplished by the sympathetic division. This is to give you an example where sympathetic division is active and accomplishes its task. On the contrary, when we are relaxed or on a vacation, or on a day off, then the sympathetic division is quiet and the parasympathetic division is active. So we are relaxed. When we are relaxed, our heart does not need to beat fast and strong, right? Our brain does not need to be vigilant and we can become sleepy, right? Our eyes do not need to be really wide open and look around. We can close our eyes. For example, if you are on a vacation in the beach, enjoying the sun, parasympathetic division is in control. Calm down, food being digested, sleepy. So this is to show you the contrast, the difference between sympathetic division and parasympathetic division in terms of physiology. So in your textbook, there is a page where you see what the sympathetic division does on each individual organ and what the parasympathetic division does on each individual organ. So I recommend you to see that and have idea how each organ is influenced by the sympathetic division and parasympathetic division. But for general knowledge, organs which have role in fight or flight situation will be stimulated, activated by the sympathetic division, and organs which do not help us to escape a danger or to fight back an enemy are inhibited. I have given you examples, right? If you want more example, for example, let's take our airway. What would happen if it is stimulated by the sympathetic division? It will be widened. It will open up because we need more oxygen when we have to run or when we have to fight, right? I already said, how about digestive system? No, it doesn't help. So it will be inhibited by the sympathetic division, but activated by the parasympathetic division. We already said that sympathetic and parasympathetic division have opposite effect on individual organs. So you can either study the table in your textbook, which gives you the effect of the sympathetic division, parasympathetic division on individual organs, or you can conclude how sympathetic division or parasympathetic division stimulus would affect individual organ on the basis of their role in fight or flight. That's how. And I told you in the beginning, each ganglion has a name, right? Especially the ones for the sympathetic division. These ones which are next to the vertebral column, besides the vertebral column, are known as together collectively sympathetic trunk. But those in front of the vertebral column are known as prevertebral ganglia, but these prevertebral ganglia are not more than 3, and their name is, the uppermost is celiac, and then below it there is superior mesenteric and inferior mesenteric. Those are the 3 ones, 3 prevertebral ganglia. So some preganglionic neurons synapse in the sympathetic trunk in this ganglia. Some pass through this ganglia without synapsing and synapse in this prevertebral ganglia, in one of them, but they do synapse either in here. Those which synapse in the prevertebral or collateral ganglia, the celiac, the superior mesenteric, the inferior mesenteric, those do not synapse in the sympathetic trunk ganglia. In those which do not synapse in the sympathetic trunk ganglia, these ones, they are known as splanchnic nerves. Splanchnic nerves, they do synapse in the prevertebral ganglia, the ones in front of the vertebra, the celiac, the superior mesenteric, and the inferior mesenteric, and they proceed to the individual organs. So I have mentioned to you about the neurotransmitters used by the sympathetic division, parasympathetic division. I have tried to show you the difference in anatomy of sympathetic and parasympathetic. You also need to know about the difference between somatic and autonomic nervous system. Okay, as we have seen, receptors on postsynaptic neurons, which are stimulated by neurotransmitters, there are also receptors on ganglionic neuron organ junctions. At the ganglionic neuron organ junctions, on these individual junctions or synapses, there are receptors on the organs which are stimulated by the neurotransmitters. There where neurotransmitter is released, there is a receptor for the neurotransmitter. So we have seen neurotransmitters on neuronal synapses, which are stimulated by the neurotransmitters. So I want you to know that there are also receptors on these neuroorganic junctions or synapses, which are stimulated by those neurotransmitters released there. What we have in front of us here is anatomy of the parasympathetic division. I told you the neurotransmitter released by ganglionic neurons at the neuroorganic junction is acetylcholine. So receptors for acetylcholine, which we find on organs, are muscarinic and nicotinic. Muscarinic and nicotinic. Actually, nicotinic receptors are on skeletal muscles, but on these internal organs, the receptors are muscarinic. And acetylcholine has excitatory effect on most of them, and what effect it has, you will find on the base of the criteria I told you earlier, how to predict what effect the stimulus of a sympathetic division or parasympathetic division has on an organ, depending on its role on fight or flight, or using the table I told you, which is in your textbook, right? So the point here is about receptors. There are receptors there where is neuronal junction or neuroorganic junction, and where neurotransmitter is released, there is a receptor for those individual neurotransmitters. So the receptors for acetylcholine on these organs is muscarinic receptor. So I want you mainly to know the presence of receptors, and the effect depends both on neurotransmitter effect on the organ, whether that organ will be stimulated or inhibited, depends on both the neurotransmitter and the receptor. The same is true about the sympathetic division. In these ganglionic neuron organ junctions, neurotransmitter is released. We said norepinephrine is released, and there are receptors for norepinephrine. Norepinephrine is a neurotransmitter released here, and there are receptors for norepinephrine. And those receptors for norepinephrine are alpha, beta, and then further they are subdivided into alpha 1, beta 1, alpha 2, beta 2. Alpha 1, alpha 2, beta 1, beta 2. So on these organs, you either get alpha 1 or alpha 2 or beta 1 or beta 2. So Alpha 1, alpha 2, beta 1, beta 2 are receptors found on these individual organs. Which receptor is on which organ, you can see in the table. And what effect, you can also see in the table, or you can conclude on the basis of their role in fight or flight situation, right? So the point I am adding here is on these individual organs are receptors which respond to the stimuli applied by the neurotransmitter norepinephrine regarding the sympathetic division. Speaking of receptors, there are also sensory receptors. And these sensory receptors are specialized cells or parts of cells which have the capacity to detect specific conditions. Sensory receptors are special cells or parts of cells which have the capacity to detect specific conditions. Those specific conditions can arise within our body, like pain, hunger, thirst, or from the external environment like heat, temperature change, touch, pressure, which come from outside, sound, vision, and so on. So sensory receptors can be divided into general and special. We can divide them on the basis of the sense they detect, general sense and special sense. The general senses which are detected by the specific receptors are pain, touch, pressure, vibration, and proprioception. Pain, touch, pressure, vibration, and proprioception. You may be new to the word proprioception. Proprioception is our ability to recognize where parts of our body are, even with closed eyes. Whether our arm is in front of us, besides us, or behind us, or whether our leg is flexed, extended. You may say, I can see that, but even if you close your eye, even if one is blind, you can still know that, and that is known as proprioception. We may not appreciate that sense unless we lose it. By the way, useful features are noticed when they are lost. When we have them, we may not notice them. So these are general senses. The special senses are the 5 special senses, the smell, taste, audition, hearing, vision, and balance. Those are special senses. So what's the difference between special senses and general senses? General senses are located all over our body. You can feel when you are touched on your forehead, you can feel when you are touched on your leg, anywhere. Those general senses are all over our body, whereas special senses are localized to specific places. For example, we can see only with our eyes, we can hear only with our ears, and so on. We can taste with our tongue, or we can smell only with our nose. So special senses are localized. In addition to that, special senses are more sophisticated structurally. General senses are much simpler. General sense receptors, for example, general sense receptors are nerve endings. Nerve endings, dendrites, can serve as receptors for touch, for temperature. In addition to that, some receptors can detect more than one item. For example, nerve endings may detect temperature change, may detect touch, may detect pain. But special senses specialize only in that stimuli for which they are adapted. So there are some characteristics of sensory receptors. And those characteristics of sensory receptors are specificity. I said receptors detect only the sense or the characteristics they are specialized for. That's one. By the way, what do these receptors do with the stimulus? When they are stimulated, receptors change the stimulus into graded potential. And if it is strong enough, it can further change into action potential. So the role of receptors is changing a stimulus into graded potential. And if the stimulus is strong enough to change the resting potential into negative 55, as we talked there, then it can become action potential and it can be felt. But if it is weak, it may not be felt. For example, if someone whispers behind you very low voice, that could be weak. It may not stimulate and cause strong enough graded potential. It may not be heard. So the role of I want to emphasize the role of sensory receptors changing stimulus into action potential, and that process of changing stimulus into action potential is known as transduction. The verb is transduce. That means they transduce a stimulus into graded potential and further into action potential if the stimulus is strong enough to change the graded potential to threshold level, which is, we said, mostly is negative 55 millivolts. We call a stimulus sensation if it is detected by our receptors. We call it perception if it has been, if we have been aware of it. What does sensation mean? What does perception mean? How come there would be stimulus and it would be changed into action potential and we won't be aware of it? By the way, if we go into the list of receptors, one of the receptors, for example, is baroreceptor, receptor which detects pressure, blood pressure within our body. By the way, there are receptors in our body which detect blood pressure, and within our organ, there are autonomous processes which monitor our blood pressure, and when our blood pressure goes high, there are mechanisms by which it can be brought down, but we are not aware of that. But our body does what it is supposed to do. We need medication when that mechanism doesn't work. So blood pressure within our body, or chemoreceptors which detect acidity, they do detect, they report to some parts of our brain like the brain stem, and there is a reaction reflex which adjusts that change into normal. We may not be aware of it. That's sensation. Perception is when we are aware of it. We are pinched, we feel it. It was detected by receptors and we are aware of it. We call that perception. Receptive field is area of the surface of the body or area of part of the body which is covered by one receptor. That's receptive field. Adaptation is one of the characteristics of receptors. What do we mean by adaptation? Adaptation is receptors stopping changing a stimulus into graded potential and action potential when they are continuously bombarded by the same magnitude of stimulus. For example, let me show you an example. When you enter into the freeway from a street, right, you notice the noise of the cars, but after you drive a mile or so, you stop noticing it. It is because your receptors are adapting, getting used to that noise, and no more transducing, no more changing the stimulus into graded potential and action potential. That's adaptation. Some receptors adapt quickly, and they call them fast adapting receptors. Some are slow to adapt, they call them slow adapting receptors. A good example of slow adapting receptor or tonic receptor is pain receptor. We call them nociceptors. Pain receptors never adapt. If you have pain, you will never ignore it. So some stimuli like noise can be ignored when our receptors get used to that noise, but Stimuli like pain are not ignored. Receptors can be classified on the basis of the type of stimulus they detect. For example, pain receptors are known as nociceptors. Temperature receptors are known as thermoreceptors. Receptors which detect chemical substance like by taste, by smell are known as chemical receptors. Mechanical stimuli like pressure, touch are known as mechanoreceptors, right? I already mentioned to you baroreceptors which detect temperature. Proprioceptors, those which detect the relative position of our body. So those are known as the proprioceptors, right? So receptors can also be classified on the basis of the stimuli they detect. So there are specific receptors in each special sense. For example, receptors for vision, photoreceptors, are in our eyes. They are cones and rods. We will see them in more detail. Receptors for sound are known as hair cells. We will see them and how they work, and so on. You can list, name, enumerate receptors for each type of stimuli detected by our body. Now let's move on to special senses. Let's start with our vision. Okay, what do we see on this picture? Let me describe the parts we can see when we look at our friend or someone in front of us, right? This dark part in the center is known as pupil. By the way, pupil can widen and can narrow. I will demonstrate that. It is a hole, by the way, and is around the pupil is iris. This iris is composed of smooth muscles which can contract and relax. These are, by the way, controlled by the autonomous nervous system despite our will, depending on the amount of light which enters our eye. This iris, which also determines the color of our eye, can some of them contract and narrow this pupil, the size of this pupil, and can also some of them relax, some of them contract. There are two types of muscles in here. I will demonstrate that on another picture. What do we have here? This is the white part is sclera, but on top of it is there is a transparent epithelial tissue which is known as conjunctiva. That's what you call conjunctivitis when this is injected and inflamed and it turns red, right? So conjunctivitis comes from the word conjunctiva. By the way, here in the pupil, in front of the pupil is cornea. Cornea is transparent tissue through without blood vessels through which light can pass, like light would pass through the window glass, right? So this is pupil. So this is what we can see. By the way, on this picture you can see that the conjunctiva is kind of peeled off. So you notice conjunctiva covers. Under the conjunctiva, this is sclera, the outermost layer of the eyeball, which is white in color. This is sclera that is covered in front of our eye by a layer known as conjunctiva. That's what we can see on this picture. Let's move on. What do we see on this picture? On this picture, we see muscles which move our eyes around. This muscle, as you can see, is known as lateral rectus. The eye we are looking here is the right eye, so it moves our right eye to the right. And this muscle is superior rectus, it moves our eye upward. So these muscles are muscles which move our eye around. Their action is listed here, so study them. Let's move on. we need to spend time on this picture. Okay, this picture gives us a very detailed image of our eyeball. What do we see here? What do we see here? I want to start first of all with the most important idea here. Our eyeball is composed of 3 layers: 1, 2, and 3. This outermost layer is known as fibrous layer, which consists of sclera here at the back and cornea here at the front. When you follow this outermost layer around, here at the back it is sclera. This is opaque layer which doesn't allow light to pass through, but this one, cornea, is transparent, allows light to pass through, completely transparent. The next middle layer is known as choroid. Also they call it vascular layer. You can see it's all over around, right? That layer here at the back, the choroid, by the way, it is called vascular layer. Here at the back it is choroid. Here in the front it becomes ciliary body and iris. I did talk about iris. Iris is a muscle around the pupil. By the way, this is a pupil. Pupil is a hole through which light passes. It's a hole. But the iris is the muscular layer around the pupil. So this vascular layer is choroid here in the back, but when we come to the front, it becomes ciliary body. These prominent parts are known as ciliary processes, and this is iris. Iris consists of circular and radially located muscles, which we will see on another picture, and it is around the pupil. It is around the pupil. This enormous layer is retina. Retina is a place where there are photoreceptors, receptors for light, receptors for vision. So that we can say is most important. So the center of the eyeball here at the back is filled with vitreous humor, semi-solid substance which keeps the shape of our eye, and it is there lifelong without change. We are born with it and we die with it. Its purpose is to keep the shape and size of our eye, and it keeps the retina compressed against the next layer, against the vascular layer, so there won't be retinal detachment, right? You might have heard about that. So this is vitreous humor, semi-solid substance, gel-like substance, which gives our eye the shape it has. Look, this is lens of our eye, and it is attached to the ciliary body or the ciliary processes. These tiny prominences are known as ciliary process. Ciliary process is part of extension of the ciliary body, which is part of the vascular layer, right? So these string-like or hair-like structures are the structures known as ciliary zonule or suspensory ligament, which attach the lens with the ciliary body, which attach the lens with the ciliary body, or ciliary processes. So this is all over around the lens. We will see on the front view of the lens. All over around the lens is attached to the ciliary body. By the way, this ciliary body is a circle around the lens, to which the lens is attached via these suspensory ligaments. What role each part has, we will talk about it. So before we move on, I want you to notice these spaces. By the way, we name this space in which we have vitreous humor as posterior segment or posterior cavity. How about the space in front of the lens here, this space, this space, and this space? We call that anterior segment or anterior cavity. Anterior cavity is divided into posterior chamber and anterior chamber. We said we have vitreous humor, which is gel-like substance here. What do we have in this space? Is this space empty? No. It is filled with aqueous humor, liquid, secreted by the ciliary body. By the way, this aqueous humor, after being secreted by this ciliary body and flowing or being released into this posterior chamber of the anterior cavity, it can flow through the pupil into the anterior chamber. What is the purpose of this aqueous humor or liquid? It provides these structures with oxygen and nutrients. It also keeps them moist, and it can also remove waste products, and it also keeps the shape of these structures. By the way, the excessive buildup, this fluid, this aqueous humor, is drained through this scleral sinus. There is a route through which this fluid is drained, but when its drainage is somehow obstructed, that's what causes optic pressure or glaucoma, if you have heard. So this is filled with aqueous humor, this is filled with vitreous humor. By the way, this zigzagged line you see here is the anterior border of this layer, the retina, and this zigzagged border is known as ora serrata. So the anterior border of the innermost layer, the third layer of the eyeball, is known as ora serrata, anterior border, this zigzagged corrugated border of the retina. I said the retina contains the receptors. You may wonder what this is. This is, by the way, here in the retina are receptors, photoreceptors, which are known as cones and rods. Cones and rods are special type of neurons which are adapted to become photoreceptors or light receptors. So this is a bunch of axons which belong to those receptors, to those neurons, to those modified specialized neurons. Their axons come together forming cranial nerve number 2, which is optic nerve, as you can see here, right? So this is a bunch of axons which are forming nerves. As you have seen, you know what a nerve is composed of, right? Picture showing that the size of the pupil can be changed. It can widen like this, or it can narrow like this. So what determines the size of the pupil, this hole? There are circular muscles and there are radially oriented muscles. When those radially oriented muscles contract, they pull on this one, then the pupil widens. Look, when these are pulled from all sides, this pupil widens. But when these ones contract, these radial muscles are pulled and the pupil narrows. And at the same time, they are telling us that sympathetic division widens our pupil when we are really scared, right? If you look at the pupil of someone who is scared, you will see the pupil widened. If they deny you, look at the pupil. If it is widened, then they are scared. If they say, no, I'm not scared, don't believe them. If it is widened, they are. So this is to show you, and to show you how narrow the pupil can be and how wide the pupil can be and what determines it. And you can see the influence of the sympathetic and parasympathetic division of the autonomous nervous system on the size of the pupil. The broader the pupil, the more light comes in. The narrower the pupil, the less light. By the way, other than being scared and relaxed, the amount of brightness, when there is very bright light, the pupil narrows down to limit the amount of light entering into our eye, so our retina is not damaged. So the size of the pupil depends on the amount of light. That's why during visual reflex, when you light a torch on someone's eye, you should observe narrowing of the pupil. If not, then these muscles or the innervation they get is not healthy. That's why you check on that. Okay, here we have a picture of the retina, the most important layer of the eyeball where receptors are located. Look at the receptors. Here are the receptors. By the way, these yellow ones are known as cones. Do you see that their shape reminds a cone here? That might help you to remember. And notice that they are rare. But the rest, these ones are known as rods. Does this remind you a rod? So what's the difference between these cones and rods? What am I saying? These cones and rods are visual receptors. What did we say receptors do? Receptors respond to stimuli. In this case, these receptors respond to light which come from objects, reflected from objects. They respond to light. The light is the stimulus they respond to. What do? They change the stimulus into graded potential, and if strong enough graded potential, then that can be changed into action potential. So that's what these do. The difference is cones are active or functional only in bright light. When light is dim, it's only rods which are functional. Cones do not function in dim light or in darkness, but rods can function in dim light and in darkness. The advantage of cones is they give us colored image and really sharp contour of objects we look at. For example, when you look in the darkness, all you might see is shade of something, right? Not clear contour or sharp image and no color. That's rods, and these are cones. So there is a chain of neurons. By the way, this is specialized neuron which has cell body, dendrites, but very modified. This is another chain of neurons where you can see there is synapse here, right? And another synapse here. This is also another group of neurons which also have synapse. They are known as ganglion cells, right? So this chain at the end has axons which come together and form the optic nerve. And this picture is to show you what an eye doctor would see when they look into your eye through your pupil. When they use a instrument which they use to look at your retina, this is what they see. Look how the retina looks like. These are blood vessels. These are the receptors. By the way, this macula lutea is part of our retina where there is high concentration of cones. In the very center, there is a part which is known as fovea centralis. There is only cones. By the way, as we go further away from this macula lutea, the number of cones decreases, the number of rods increases as we go further away to the periphery. So most images formed on our eye are aimed or destined to be formed on this macula lutea, so we can see the best sharp image we may. This is optic disc. Optic disc is part of the retina where blood vessels enter and exit and where the optic nerve exits. So here at this circle there are no receptors, so this is considered blind spot because of lack of receptors, and because here is where the blood vessels enter and exit, where the optic nerve exists. So I want you to remember that optic disc is considered blind spot, whereas this is considered the site on the retina where best image is formed because of high concentration of cones, those which are responsible for sharp image and colored image. Okay, so here we have a closer look at the lens and the pupil. By the way, this is the iris where we have those circular and radially oriented muscles which determine the size of the pupil. Here we have the lens. I want you to notice that these suspensory ligaments or ciliary zonule attach our lens to our ciliary processes or ciliary body. So how does that function? How does that function? I will demonstrate that on another picture. By the way, this arrow is showing us the direction of flow of aqueous humor, which is secreted by this ciliary body, which I described earlier, and is showing us where it drains normally. So venous sinus is there through which this aqueous humor drains. I want to talk about refraction. Look, refraction, we need to get familiar with the concept of refraction. Refraction is change of direction of light ray when it passes from one medium to another medium. What do we mean by medium? For example, from air into seawater, right? Air is one medium, water is another medium. Or from air into our cornea or from our cornea into our lens. Look at, let's follow this light ray. This is a light ray which is arising from this point, right? When it hits the lens, look how it is bent. It changed direction. That change of direction, which happens when light ray enters from one medium to another, is refraction. Refraction is very important for our vision. The purpose of the lens, the work of the lens, the function of the lens is refracting light, so those lights can be focused on macula lutea, the site which I showed you has highest concentration of receptors. So we may have sharp image, colored image, right? So any light which comes into our eye, reflected from any object we are looking at, should be refracted, its direction changed, and sent to the retina, specifically to the macula lutea, so we may see the best image possible. So the function of our lens is refracting. So the amount of refraction needed for an object which is coming from for light which is coming from an object which is close to our eye and for an object which is far away from our eye is different. The one which is closer to our eye needs more refraction, more change of direction of light. So in that case, when we see something close to our eye, our lens thickens to cause more bending of light, more change of direction. Where when we look far away, less refraction is needed because the light rays from the object which is far away come parallel to each other. They don't need much change of direction. So the lens can flatten out. How the lens flattens out or thickens, I will show you on the following picture. But what our lens does is become thicker. By thicker, I mean its antero-posterior dimension. When it gets thicker when we look at something close to our eye, and it gets more flat and thinner when we look at objects far away. Why? Because objects close to our eye need more change of direction. And thicker lens causes more change of direction of light ray. Let me show you on the following picture how our lens thickens and flattens. Look, this is the ciliary body, and these white lines are the suspensory ligaments which attach the lens to the ciliary body. So let me ask you a question. When this muscle, with this circular muscle, which is within the ciliary body, by the way, they are also known as ciliary muscles, when they contract and when this circle gets narrower, what do you think will happen to our lens? Our lens will be thicker. So to understand that, let's consider the opposite. Let's say this circular muscle relaxes and the circle becomes wider. When that happens, these ligaments become tense and they pull the lens from all directions, forcing it to flatten out and become thin, thin, T-H-I-N, in its anterior-posterior direction. So we will have more flat, thinner lens. That is good when we look far away. But when we look close, this muscle contracts and this circle becomes smaller, and these suspensory ligaments become less tight. They become loose, and then the lens gets to become less flat, more thicker, and bigger dimension, anterior posterior dimension, which has the ability to cause more refraction, and that refraction gives the opportunity the image of the object from which those light rays came to be focused on our retina, specifically on macula lutea, where there are really high concentration of receptors, so we may see colored and clear, sharp image. So this is a mechanism which makes our lens flat and thick. So as we age, our lens loses its flexibility, its ability to flatten out, and its ability to thicken back. It loses that capacity gradually. That's why we need eyeglass when we age. So our eyeglass is playing the role of our lens. That's why sometimes eyeglasses have binocular. One part of the eyeglass good for close vision, for reading, and one for far vision, right? So because our lens loses its flexibility to change its thickness and dimension. So that is about the change. Okay, this is showing you the routes through which the action potential generated by receptors in the eyeball move. Do you remember? This is view from below. This is, we are looking at the brain from below, as you can see here, from below, inferior view. So this is optic nerve from one of our eyes, another optic nerve from our eye, another eye. So these are tracts. Didn't we say a bunch of axons running in the central nervous system are known as tracts? But a bunch of axons in the peripheral nervous system are nerves. So these tracts are leading to the occipital lobe. So occipital lobe is the center where whatever we see is interpreted. So what do you think would happen to someone who has intact eyeball, but the occipital lobe is destroyed, let's say by stroke? Will be blind. Without occipital lobe, no vision. Occipital lobe is the center of integration or interpretation for vision. We will see which center does what. We have seen this when we discussed the brain. So this is all I have about our vision. I want to bring your attention to this picture. Look at this. This is lacrimal gland, which is located on the lateral upper corner of our orbital socket. So this gland produces tear, which drains through these ducts into the eyeball. By the way, this gland produces tear continuously. We don't notice it because normally it produces in a small amount, and that drains through lacrimal punct and lacrimal canal and nasolacrimal duct into our nose, where it is absorbed in a small amount. But when we cry, when this gland produces more tear, then it starts to flow out. And we need to clean our nose. Do you know why we need to clean our nose when we cry? Because the amount of tear going into our nose is more than what can be absorbed. So it overflows. So what is the purpose of tear? Tear keeps our eye moist so moisture is needed. Lubricates, so there is no friction between our eyelid and eyeball. It also prevents infection because it has antibacterial effects, and it also kind of nourishes this surface, washes it off. Any dirt is washed off. When the eyelash even goes in, it can be washed off with tear. More tear is produced and it is washed off. So this is the role of the lacrimal gland, which produces tear. Our ear in general does two functions: hearing and balance or equilibrium of our body. The two functions are done by our ear: hearing sound to sound and equilibrium, balance of our body. So in general, our ear has three parts. This is the external ear. Here is external ear, which includes the pinna, lobes of our ear, and external acoustic meatus or acoustic canal. Starting from the tympanic membrane, it is middle ear. This part is middle ear. This part, let me circle with my cursor. This is middle ear. Tympanic membrane is the border between external ear and middle ear. Middle ear is this cavity where we have these three tiny bones. The name of these bones is malleus, incus, and stapes. To remember the names, does this remind you the step on which you have to step to get on a horseback? Malleus means hammer. Does this remind you hammer? Incus is part which metal workers use to put something on and hit with a hammer, right? So the next part is inner ear. So we said external ear, middle ear, and inner ear. By the way, as part of the middle ear, I want you to notice this acoustic meatus or auditory tube or pharyngotympanic tube, which leads to our mouth, by the way. This auditory meatus serves to equalize the purpose of this auditory meatus, or the function of this auditory meatus is to equalize the pressure in here and outside in the atmospheric air. So the tympanic membrane is exposed to the same pressure on both sides. Through our mouth comes atmospheric pressure here, and through the external ear comes atmospheric pressure here. So the tympanic membrane has the same atmospheric pressure on both sides. That is why you are advised to keep your mouth open when you fly on an aeroplane as the aeroplane increases altitude, because pressure changes. If you keep your mouth closed, you have the pressure from the ground on Earth here and the new pressure here, so imbalance between the two sides. But when you open, you will have the new pressure here and the new pressure here. The new pressure as altitude increases on aeroplane. So this is inner ear. Inner ear houses the semicircular canals, three of them. There are three of them, these semicircular canals: the vestibule and the cochlea. This cochlea, this spiral 2.5 coiled tube, is responsible for sound hearing. This part, the semicircular canal and the vestibule, are responsible for hearing. What you see here is cranial nerve number 8, vestibulocochlear nerve, leading to the temporal lobe of our brain. So this is to see the whole ear. Let's move on to a more detailed picture. Here we have the middle and inner ear in bigger size, in more detail. Tympanic membrane, so the external ear is left here, right? And the ossicles and the inner ear with the semicircular canal, two of them visible. the semicircular canal are three, in the three planes. These semicircular canals are located in the sagittal plane, in the transverse plane, and in the frontal plane. They are perpendicular to each other, three of them. This is a 2.5 coiled canal. I'm going to talk more. Here is the eustachian tube or pharyngotympanic tube, auditory meatus. It has three names, which communicates the middle ear to the mouth. By the way, the reason why children develop middle ear infection when they have common cold is because infection comes through this and gets in here. That's why they develop. That's one of the reasons. The other reason is because their defense mechanism is not strong enough yet. But in adults, this canal becomes coiled. In children, it's straight. So it's easy for infection to ascend into this middle ear, but in adults it is bent and is not easy, and long. In children, straight and short. In adults, bent and long, and it's not easy for bacteria to ascend. And in addition to that, in adults the defense mechanism is already mature. Let's move on to another picture. Middle ear alone shown here, so you see all the bones clearly. Here is the inner ear. We are going to spend most of our time on the inner ear. Did you remember I told you that the semicircular canals are three in number and they are perpendicular to each other? This is the one in the transverse plane. They call it lateral, the semicircular canal. This is the one which is in the sagittal plane. They call it anterior. And this is the one in the frontal plane. They call it posterior. They are perpendicular to each other and they are in that position for a reason. Let me tell you the structure of the inner ear. The inner ear is composed of two tubes, one on top of the other. The outer tube is bony tube. They call it bony labyrinth. It is composed of bone. And the inner is membranous labyrinth, and it is composed of soft tissue membrane. So two tubes, one inside the other. That is not only in the semicircular canal, it is true in the vestibule, it is true in the cochlea. By the way, this coiled part, which is known as cochlea, is responsible for our listening, audition. This vestibule and the semicircular canal are responsible for our balance. And we will see one by one how they do that function. So, in this membranous canal is fluid known as endolymph, and in between the membranous canal and the bony canal is fluid known as perilymph. Endolymph in here, perilymph here, in between the two canals. The same is true everywhere everywhere in the inner ear. So this is to give you these nerves arise from all parts of the inner ear. This is finally becomes vestibulocochlear nerve, which is cranial nerve number 8, and it leads some part of it lead to the temporal lobe of the brain, and the other part to the cerebellum. So it has two parts: vestibular branch and cochlear branch. They unite to become vestibulocochlear. That's why its name consists of two parts. So let's move on to another picture to see more detail. Okay, let's take this one. Okay, look, this is the cochlea. Now we are dealing with the cochlea. The part I said, the 2.5 coil, which is responsible for hearing. If you cut through the cochlea, do you see the section taken through the cochlea? This is what you get. So if you take one of these, do you see this rectangle or square here? When you magnify, this is what you see. Didn't I say there are two canals, one inside the other? This is the inner canal and this is the outer canal. So when you zoom on the inner canal, which is known as cochlear canal or cochlea, this is what you would get. So this is where we have receptors for sound. Let me take you to a bigger picture so we see the receptors, but I want you to see the big picture. We made a cut through the cochlea, which is a 2.5 coil. We got this, and then from here we took one of the rounds, one of the canals, which is this, and within this we took the part which is known as organ of Corti. Organ of Corti is receptors for hearing, for sound, plus supporting cells. By the way, receptors for sound are hair cells. Why do we call them hair? Because at one end, at the upper end, they have hair-like structure, which bends when stimulated by sound, and which makes the hair cell transduce or change that stimulus into graded potential and further action potential if strong enough, right? So these are the hair cells, 1, 2, 3 here, and some more here. So these are hair cells. The other are supporting cells, which have assisting role. So together with the cells which have assisting role, the support cells, the whole structure is known as organ of Corti, but the receptors are the hair cells, these ones. This is supporting structure. It is known as tectorial membrane, which is lying on the hairs of these hair cells. These cells are known as hair cells because they have hair on them. And the bending of the hairs is strong enough stimulus to make these hair cells change sound wave into graded potential. Let's have a closer look on this organ of Corti. Don't forget from where this organ of Corti is taken. Here we have the organ of Corti. Here are the hair cells. Do you notice the hairs now? These are the hairs. They call them hairs or stereocoli. So when sound comes and stimulates them, it is these hairs which bend, right? And they generate a graded potential, strong enough action potential, which will be propagated along these nerves. This is part of cranial nerve number 8. Cranial nerve number 8. Let's move on to another picture. Look, this is showing us if the cochlea, which is into a 2.5 coil, could be straightened, it would look like this. This is how it would look. So this is a good picture. I want you to really pay attention to this picture. This picture talks a lot. The reason why we straighten the cochlea, which is a 2.5 fold, is so we understand how sound wave stimulates the receptors in here and convert that sound wave into graded potential and action potential. Look, when sound arrives into our ear and hits the tympanic membrane, as tympanic membrane moves, the malleus, which is attached to the tympanic membrane, moves, and that moves the incus and steps. As these are moved, this sound which hits the tympanic membrane is amplified. So these small bones vibrate. As these small bones vibrate, the steps pushes on the oval window. This is oval window. This steps is attached to the oval window. There are two windows. Here is a round window and there is a oval window which is attached to scala vestibuli. By the way, this upper side is scala vestibuli, this lower side is scala tympani. Scala tympani, as you can see labeled here, right? So this oval window is directly connected to the scala vestibuli. Scala vestibuli is just a canal where there is perilymph, and on this side, this round window is there, which kind of equalizes the pressure. So when the steps pushes against the oval window, the fluid in here, the perilymph, is moved, right? So when that happens, don't forget that the receptors for sound are located in here. Depending on the frequency, of the sound, the sound wave or the pressure will bend that part of the cochlea which is responsible or which responds to that frequency. Some will bend here, some will bend here, some will bend here, depending on the frequency. So whether sound is from piano or from guitar, from Mr. A or Mrs. X, that is determined, the frequency determines and is determined by the part of the cochlea, this inner cochlear tube, which is bent. Some could be bent, could bend this part, some could bend this part. So that determines how strongly this tube is bent. Also determines the volume of the sound. The stronger the volume, the more the tube is bent. So as I told you, the hair cells are inside here, and when this is bent, those hair cells respond to the bending caused by the pressure, and those hair cells convert this pressure, this vibration, into graded potential, if strong enough, into action potential, and that is transmitted through cranial nerve number 8 to the temporal lobe, where it is interpreted and we understand what we heard. So what am I telling you here? Sound wave will vibrate the tympanic membrane, and that will be amplified by these ossicles or tiny bones. And these steps, the last in the chain of the tiny bones, will transmit the pressure into this fluid-filled canal, which is vestibular tube, and this pressure or this vibration will stimulate a specific part of this inner canal in which the receptors are. Depending which part of this canal is stimulated or responded, that determines what type of sound. Is it an instrument? Which instrument? Is it Mr. X? Is it Mrs. Y? Or any other body? That depends on which part is stimulated, which part responded to that vibration, and how loud, how much that part is bent, how strongly it is bent. The stronger it is bent, the louder the voice. So, and then when that happens, the hair cells which are located inside here will transduce the sound wave into graded potential, and then that is transmitted through cranial nerve number 8 to the temporal lobe. Temporal lobe plays the role of integration or interpretation of the sound, and that results in what we hear. This is more or less how we hear sound. Let's continue. Okay, the vestibuli and the semicircular canal. I said the semicircular canal and vestibuli are responsible for our balance. How do they help us with our balance? They tell us when we kind of lose balance. They inform us when we kind of lose balance. So we have here 3 semicircular canals. We have here 2 vestibular parts. This is saccule, this is utricle. What do they do? Let's start with the utricle and saccule. By the way, the utricle and saccule are responsible for linear motion, meaning motion in straight line. When we move, for example, in a car, when you step on the gas pedal and accelerate the speed of your car, these receptors, by the way, the receptors again here in the vestibuli, in the saccule and utricle, are again hair cells, but together with supporting cells, the whole apparatus, together with the hair cell plus the supporting cells, is known as macula. And this is a vestibule. By the way, The reason why this is called vestibule is so you remember it. You know that if you start from the vestibule of a building, you can go into any room going through the vestibule, right? This also will allow you to go either to the cochlea, this part, or to the semicircular canal. That's why they call it vestibule, I guess. So here in the vestibule, two chambers. One is known as saccule, the other is known as utricle. Both of them are responsible for linear motion. They keep us informed about linear motion. Look, when you step on the gas pedal, or if the driver steps on the gas pedal and you are on the driver's seat, you would incline backwards, right? That incline backwards, like the lady down here, will cause these hair cells to bend, and that will be transduced into graded potential and further to action potential, which we will feel and react accordingly if we are going to fall down, right? We prevent ourselves from falling. Or if suddenly the driver steps on the brake, right? This is what might happen. You would be inclined forwards. You may stretch your arm against the dashboard so you don't slam against the dashboard, right? When that happens. So this is how linear motion is detected namely by the utricle. So receptors are again hair cells, but together with the remaining structure which have assisting role is known as macula. How about the saccule? What does the saccule do? The acceleration, the horizontal movement, which I explained while you are in a car, either sudden stop by stepping on the brake, with this kind of changes in movement is detected by the utricle. I mean, utricle detects the changes you would see when a car is accelerated and you are moving horizontally, or when the driver steps on the on the brake, right? So horizontal movement, accelerated movement, or sudden stop or deceleration, it is detected by the utricle with this kind of changes in the receptors, right? That is done by utricle. What does saccule do? Saccule does or detects the changes which we may feel either when we ascend in an elevator upwards or downwards. That is because of the position. The receptors and the receptor apparatus is the same thing, macule also here, but the position is different. Because of that, this one, the saccule, detects or ascending upwards or descending downwards, depending on the force of gravity on this structure. So when aeroplane increases altitude after a takeoff, or when it decreases altitude during landing, that sensation is detected by the saccule, whereas utricle detects horizontal movements like accelerating the speed of a car while you are sitting in it, or sudden stop. You may feel that if you are in a passenger seat. So that is about these vestibules. Linear change in motion. Linear change in motion. Horizontal by the utricle, vertical by the saccule. So saccule detects the kind of movement which we feel when we ascend on an elevator or on a plane, or when we descend again on an elevator or on a landing aeroplane. How about semicircular canal? For that, let me take you to another picture. Look at this. Look what this ballet dancer is doing. When she rotates in here, this rotation will cause fluid to move in the fluid, the perilymph and endolymph in here will be moving, right? That will move the hair cell and we will feel that. That is felt. So semicircular canals detect rotational movements. The saccule and utricle, straight line movements, but semicircular canals rotational movement. This is detected by the semicircular canal, which is the lateral one, which is in transverse plane. What about if we do acrobat, right? Tumbling or flipping, that would be detected by the medial semicircular canal, which is in the sagittal plane, and so on. Depending what type of rotation you do, that will cause the hair of the hair cells to be bent. By the way, the receptors at the base of the semicircular canals is known as ampulla. It is known as crista ampullaris, right? Again, hair cells are receptors together with the assisting structures. It is known as crista ampullaris, and depending what type of rotational movement we do, the hair cells will be bent. That bending will be converted into graded potential, further into action potential, and through the ACE, the vestibular branch of the vestibulocochlear nerve, is transmitted to the cerebellum and interpreted there. So in the vestibule, the ventricles detect horizontal motion, accelerated car or suddenly stopped car. The saccule detects ascending and descending like in an elevator and aeroplane. Semicircular canals detect rotational movement. In all of them, receptors are hair cells. This is all I have for today.